Okay.
Good afternoon from Monterey. My name is Jean Dupré from the James Martin Center for Non-Proliferation Studies, and I'm pleased to welcome you to this CNS webinar. It is with great pleasure that I'd like to introduce Dr. William Potter, the founding director of the James Martin Center for Non-Proliferation Studies and Middlebury Institute professor, who will introduce our speaker. Thank you both.
Thank you very much, John. You know, in the 30 years since I founded the center, I've shared innumerable seminars and meetings, but this is the first one that I've had the pleasure to welcome folks virtually to a noontime CNS seminar. So it is a pleasure to welcome you all to this webinar this afternoon on a topic that couldn't be more timely. We also could not have a more knowledgeable expert to address the issue of late-breaking COVID-19 developments and the implications than our own director of the CNS CBW Non-Proliferation Program, Dr. Richard Pilch. A physician by training, Dr. Pilch has been associated with the center since 2002 when he completed a postdoctoral fellowship here. Following his initial stay in Monterey, he spent nearly a decade overseas dealing with biological security issues, including those posed by the former Soviet Union's biological weapons program. During this period, he was actively engaged in training scientists from many countries on non-proliferation, on bioethics, and also global health security, and planned and also implemented literally dozens of cooperative research projects and pandemic response exercises across the former Soviet Union. Dr. Pilch has led multiple threat reduction programs on behalf of the US government, served on over 30 technical panels and advisory boards, and has authored more than 60 publications of one sort or another, including the authoritative encyclopedia of bioterrorism defense with his longtime mentor and former CBW Non-Proliferation Program Director, Ray Zelinskas, which came out back in 2005. Rich rejoined CNS last year as director of our CBW Non-Proliferation Program, and we're very fortunate to have him as the main face of the center during our current horrific COVID-19 pandemic. One of his innovations was the launch of the World War V portal on the CNS website, which provides analyses of a wide range of pandemic issues, as well as Q&A opportunities in which Rich and other CNS non-resident scholars can respond to inquiries from the general public. Earlier this week, for example, Rich and former Assistant Secretary of Defense for WMD, Andy Weber, a member of the CNS board, published an important analysis on the World War V site about Kazakhstan's national biolaboratory, which some Russian and Chinese sources had falsely claimed was engaged in offensive biological weapons work. So I can say a great deal more about Rich, but I think the time would be better spent having him speak about recent developments related to COVID-19. So I'm pleased to turn the virtual floor over to you, Rich. Look forward to your remarks.
Thanks. Hi, everybody. I hope you all can hear me well. I'm going to share my screen now just to get the slides up. Okay. Most important couple points up front. This was originally prepared as a follow-up presentation to a briefing I gave to the CNS Visiting Fellows about 2 months ago. CNS has fellows in professions other than biology that come to the center for periods of time and work with our scientists and experts in their fields. We have 5 visiting fellows: Anastasia number 2, Hannah, Fadime, and Rabia. They are now stuck with us in the US and riding out the storm with us. So I'd like to extend my my welcome to them. The original focus of my last talk was really international because they were the audience, but this will be a very US-centric response briefing. So the second thing I'd like to just mention, I put a picture of my girls up there in the corner. They helped me with preparing this PowerPoint, especially the staging of the slides. So for the layouts, they get due credit. Just as an example of where we all are in the current pandemic, that picture you see there of us in the Jeep, we just had to put balloons on the Jeep and go do our first drive-through birthday party, where the plan was actually to throw the present out the top of the Jeep to my older daughter Aisha's friend, but because it was raining, we had to put the top up, and our new plan was to extend it out of our window with a golf club. The reason we could throw it out of the top of the Jeep was because it was actually like a pillow of a poop emoji. So normally I wouldn't recommend throwing presents out of a Jeep as you're like whizzing by someone's house. But and sorry, Rhea, if I just gave away what your present is, but happy birthday from Aisha, and thanks to my girls for helping me put this briefing together. I think those 2 points bring me to my third and final point on this first slide, which is, we're going to be talking about real people. That's what this all comes down to. I'll be going through numbers, this many deaths, this many infected. If we do this, this many people might die. These are real people, okay? And I know you all know that, but it just needs to be said because I may forget to emphasize that point. I may be talking about, the essential workers that are out there keeping this country going, and I may be referring to them in the same way we might refer to a canary in a coal mine and saying we need to look at these people and understand what they're being exposed to. And that will come across wrong. And the point is, these are real people being affected and real people we need to protect, but they're also our greatest asset, obviously. So let's just keep that in clear going in. So almost all of my thinking right now is trying to figure out what is our end game? How do we get through this together? That will be the focus of this presentation, but as a lead in, I'll start with just one slide on where are we now. Then I'll do just a couple slides on how we got here. And then we'll go through what I think the different scenarios are moving forward, and what factors may play into those. Again, as Jean mentioned, if there's a question related to an individual slide, if you type it, I will try to remember to check the Q&A after I move on to every slide. And if it's relevant to the slide, I'll try to answer it. But since we have about 15 slides to go through and there's just so much information and so many rat holes I could end up going down, I don't want to distract too much. So we may end up reserving most questions until the very end. Okay, so next slide. Where are we now? So this is a busy slide. I apologize a little bit for that, but I like to see sort of all the details in one place. And what we'll do is we'll sort of walk clockwise through the pictures. We're over 5 million cases now, and the US has by far the most, 5 times more than the next highest country. We also have by far the most deaths. Sorry, this is from Johns Hopkins website, which is what I personally use to track the data. So it's a great resource. The link is there if anyone wants to follow it. If you look in the lower right part of the top image, you'll see a curve. And the key point about this curve, that is a global curve. But the key point about this curve is this is just starting. So anyone that thinks, okay, we're almost through, with apologies, that's just not true. If you move to the curve below that, now a blue curve, now that's just the US curve. Really, the US curve is driving the international curve right now because we have the most cases. But if you look, our curve has gone up and it just sort of keeps going, right? It's not coming back down, but it's not going up further. That's what we would call a plateau. So if we want to predict where the curve is going, there are really 2 factors. How many people can still get infected and how much exposure they get where they will be infected. So those are the 2 key drivers. If you start your response with a very weak social distancing approach, which is essentially what we've done, we've left it to the states, we haven't had a coordinated national response. So we're seeing sort of continued exposures. If you think about susceptible people as, forgive me, but acceptable people as sort of a gas, and the virus as the flame. We sort of are just pouring gas on the flame at a lower level, more in some areas, lower in other areas, and we're seeing sort of this extended plateau. Now, the alternative, if you were to look at, say, a curve like the CS-only curve for a country like South Korea, is you would see it go up and whoop, come back down. That's because they actually did a very strong initial social distancing. They used tons of testing, very responsive social distancing and contact tracing, and they were able to effectively suppress the caseload. So the caution with that approach is that they are now set up for a bigger curve later if they make a mistake, right? So again, we're talking about susceptibles and exposure. In our approach, we're getting rid of some of our susceptibles and we're getting rid of some of our exposures. We're finding a lower baseline to sort of ride this out. In South Korea's approach, they're not worried about susceptibles. They're trying to get rid of exposure. So if they ramp back up their exposure, they've got a big susceptible population, and we could see a big burst in cases in a country like South Korea or in China, where they took more of a sledgehammer approach to knocking down the exposure to the virus. So if there's a silver lining in all of this, it's that our sort of balanced approach to reducing susceptibles as well as the potential for exposure, it's preserved our healthcare capacity to some extent. It's given us an opportunity to get ahead of the virus to some extent, which we've sort of missed our opportunity on that, to be frank, but to some extent. But almost most importantly, we've had sort of this leaky lockdown where we continue to slowly have a decline in susceptibles. We continue to slowly build our herd immunity, and hopefully, unless we do something, very non-scientific, we should be preserved or spared a large peak later, like those that we've seen in past pandemics. And I'll sort of go into those scenarios a little bit later. Okay, so I don't want to spend long on how we got here. We all went through this together, but since it is the most common question I get, when I'm asked about the pandemic, I'm asked, how did this happen? And, my answer is, really the most common question is, what could we have done to keep this from happening? And my answer was, well, we let it happen in the first place. We knew, and we've known for a very long time that human practices causing ecological disruption are increasing our interface with a whole number of viruses we've never experienced, and many of them are going to cause problems, right? But there's been no international initiative to address that problem. You'll see some initiatives on climate change, and this is very similar, but really no focused effort to reduce both the supply and demand of the human behavioral practices that bring people in contact with the animals that hold the viruses that can cause these types of pandemics. So that's the biggest thing. That's the biggest thing that caused all of this. We didn't address that. Now, if this actually turned out to have come from a laboratory accident, which I'll talk about in the next slide, we also knew that was a problem. We knew there was global proliferation of numerous laboratories without any sort of global safety process, global accreditation process. And we also knew that a lot of these laboratories were performing high-risk experiments that could lead to this type of pandemic if there were an accident. High-risk experiments, not biological weapons, very clearly not biological weapons, but high-risk experiments that I'll go into a little bit later to try to understand what the potential is of all these millions of viruses out there that we're starting to identify. So both of those problems moving forward, if there's anything I can sort of convey with this brief, we have to address those problems at an international level to make sure this never happens again. So the second thing about how we got here is let's give the virus its role in this. This is a very adept virus. It spreads as early as 2 days post-infection. So typically, a normal surveillance program, you would watch. If someone gets a fever, you pull them out, right? That's why we started checking fevers at businesses and things like that. Here, 2 days after someone gets, as early as 2 days after someone gets infected, so it could be later, but as early as 2 days after someone gets infected, they can start shedding virus, which means other people can start being infected, having no idea that they're doing it. That's very hard to control using typical epidemiological and public health practices. Second, a lot of the people who become ultimately infected don't ever get symptoms. So now we can extend that period where people who don't know they're infected are able to continue to shed the virus. Third, the virus appears to shed the most at the very beginning of the infection, which is shocking. In fact, maybe it's not shocking to people that do this all the time, but to me, I can remember I had a slide I had a weekend workshop right before this was all starting, and I had a slide that said, I think that as long as we control looking for fevers, we should be fine, because what we learned from SARS was, the original SARS in 2003 was your viral shedding from an individual person that enables transmission to another person increases over time. So that the reason SARS 2003, it didn't end up being something huge, was most people who were able to transmit the virus were already in intensive care in hospitals. So you saw the majority of people becoming exposed were actually the caretakers in the hospitals working with them. That was when they were doing the most shedding, and that was when people were most at risk of contacting the virus. Here it's flipped. We're having the highest risk right up front when we barely know what's going on. And keep in mind, this is all well before social distancing was in place. And there were, after the initial identification of the virus in China, there were at least, I think, 1,300 flights between there and the US, direct flights with over 40,000 passengers, seeding the virus into our population unintentionally, right? But seeding the virus into our population. Even more index cases than from China came from Europe, where the flights continued. And people were traveling thinking, I think most people would have been responsible and said, hey, if I have a fever, I probably shouldn't go. If I have a cough, I probably shouldn't go. If I'm short of breath, I probably shouldn't go. But a lot of these things came from people who had no idea, or they were incubating the virus and developed symptoms later. So the virus needs its due in understanding how we got here. All that being said, we did miss an opportunity. The bottom line is, cases would have come to America anyway, but I'll just use myself as an example as what maybe happened with us. So for me, I'm supposed to understand this stuff or pay attention to it, right? And I saw cases starting in China of a new virus, and my mind was, this is normal. Every year in the same location, at the same time of year, we see new cases of something. Typically, it's a new type of influenza. Remember, that's where H5N1 influenza came from, H7N9 influenza came from, of course, the original SARS. And so, you're sort of desensitized to it after a while, and you think, okay, another thing happening in China in winter, no insult to my Chinese colleagues, but another thing happening. And you sort of just assume, hey, it's more of this ongoing ecological disruption and activity at the animal-human interface that we talked about earlier. And, sort of fall asleep at the switch. For me, it didn't become real until very specifically February 21st. And the reason it became real on February 21st was because I was supposed to fly to Milan the next week. And that was the day that it went from 4 cases in all of Italy to, by that February 21st was a Saturday, by that Monday, there were 500 cases and my trip was canceled and my conference was canceled. And I was realizing, hey, this thing broke containment, emergency. It's coming to America if it's not already here. And of course, we've now found out it's been here for a long time. So I think, first, we sort of lacked that initiative. Second, in fairness to everyone involved, we've never really known who was in charge. Initially, it was the head of Health and Human Services. Then it became Bob Kadlec from ASPR, the Assistant Secretary for Preparedness and Response, who's outstanding. Then it became Vice President Pence. We always see Tony Fauci. we see the president, but it's unclear messaging and some diversion to states and some US government telling us what to do. And there's never been a clear communication coordination, obviously. But then getting to the third point and the biggest point, we didn't do enough testing. Again, the countries that had the most successful response, test. If you test, you find out where the cases are, you isolate them, you identify their contacts, you quarantine them, and that's how you take a scalpel to a problem like this, as opposed to a sledgehammer, which is everyone stay at home, right? There are reasons other than we had a faulty test that caused the problem. First, we didn't have a broad enough case definition. So case definition will tell you who gets tested. And initially we required people to have had contact or contact with a known case or travel from China. Then eventually we extended that to contact with a known case or travel from China or affected areas of Europe. That's to even get a test. Well, by then it's too late. What we really need to do was just start testing everybody. And I'll have a whole nother slide on that later. So we sort of missed our opportunity up front. And then I'll just add a bullet here now that I'm not saying we've blown it with our current shelter in place, because the point of shelter in place to a large degree, at least the stated point, was to keep our ICU healthcare capacity from overflowing. And in some areas that's been successful and some arguably not, right? Particularly in New York City. But I was really hoping we would use this time to get out in front of the virus, get testing out, get contact tracing out, so that we can adopt a more surgical approach. And we don't appear to have done that. With the ancillary benefit of the leakiness that may be preventing a later spike. All right, so I'm done with this slide, and I think I got a question, so let me see if I can pop that up. Oh, yeah, I'll jump into both of those questions at the end. Okay, thanks. All right, so we're still on how we got here, and I'll be honest, I was really hesitant to go into this this slide because I'm really not an expert, but it is, after what went wrong, this is always the second question I got. Where did this virus come from? So let me talk you through it as I understand it in simple terms, because that's the only way I can. So from a descriptive epidemiological standpoint, which is who, what, when, where of an outbreak, which is your typical outbreak investigation. SARS-CoV-2, COVID-19 made a lot of sense. Like I said, it was in a place in the world where the animal-human interface has led to similar events at a time of year when we would have expected it to happen. And there just wasn't a lot that looked abnormal about that. But when you get to why and how, which is sort of the analytical epidemiological component, there's a real lack of understanding of how this thing came out, first being identified in humans already really well able to transmit from person to person. So the analogy I would use to that is if you've ever worked with someone and tracked changes, right? So you take a document and you write it, right? And then it goes around to a bunch of other people to insert their comments or updates and track changes. And you'll see the document in the original form, and then you'll see a whole bunch of things that are different, right? A whole bunch of things that have changed that are presumably improve the final product, right? And then those changes that make the product better stay. And those changes that don't make the product better go. Okay, so a virus is the same way, so anytime, so for basics, okay, and I know you guys all know this, but just so I can talk through it myself. So the most basic thing is a virus is basically nucleic acid, genomic material, genetic material, and proteins, right? It needs to be inside a cell to produce more of that nucleic acid and more of those proteins, right? So that's why we know that this virus is either in an animal, in a human, or on the surfaces around them for a very short time, right? Which is why shelter in place is supposed to work, which is why all of our mitigation things are supposed to work. Anytime, whether it's a human or a virus, you need to make more of your genetic material. You're reproducing more of the same strain through a scientific process, and you make mistakes. It's normal. All right. If you're a human, you have a proofreading mechanism that goes through and fixes those mistakes. If you're a virus, you also do. But if you're an RNA virus, you have a notoriously bad one. So even though, and SARS-CoV-2 is an RNA virus, okay? It's a virus made of ribonucleic acid. So, even though SARS-CoV-2 is actually a little bit better at proofreading, or actually quite a bit better at proofreading than other RNA viruses, it still makes mistakes when it's making copies of its own genetic material. And it's a virus, it's making tons of copies. So it's making tons of mistakes, okay? Most of those mistakes don't matter, they don't affect the virus at all. Some of those mistakes, what we would call non-synonymous mutations, will actually change the characteristics of the virus, okay? And we're talking, in a single person. In me, if I'm infected and you take a sample, I've got a bunch of different versions of the same virus because of these little mutations, okay? And so multiply that by everyone in the world who's being infected, and you've just got all these changes going on. Some of them make a difference, but the ones that propagate, the ones that become the next big thing we see, are the ones where that difference gives them a selective advantage, okay? So a typical selective advantage would be something that spreads better than the predecessor. Think about weeds on a lawn, okay? You've got normal grass, and then you've got a weed. That weed can grow faster. So it pulls all the nutrients from the soil. Maybe, I'm not a gardener, but I'm just using the analogy, pulls all the nutrients for the soil, and it overruns the regular grass, right? So in this case, the nutrients, unfortunately, are people, right? So for this particular virus, the most important part of the genome, the genetic material, is the part that codes for a protein called the S protein. S protein is where coronavirus gets its name. If you look at that upper right picture, coronas means crown, and the S proteins are all those things sticking out. Why do they stick out? Because that's what attaches to cells. And in particular, it attaches to cells with something called an ACE2 receptor. And that ACE2 receptor is predominantly located in the respiratory tract as well as other locations. So everything comes down to the gene coding for the S protein that enables binding. Now, what we know about SARS-CoV-2 that's different from SARS, the original SARS, and other coronaviruses is it binds really, really well. And so the question is, how does it do that? So let's talk through a little bit of the second giant graph there, which I know is confusing to me too, but I'll try to make it just make sense for the only point we're trying to make. Now, where did this virus come from? Okay. If you look at 0 to 100, where it says nucleotide identity, 100 equals 100% the same as this virus. Okay, so imagine a line at that 100% that goes across the top. And that is SARS-CoV-2. Now, a scientist at the Wuhan Institute of Virology found a sequence that was damn close to that. Sorry, my kids might watch this, so very close to that. Sorry, girls. And that line runs, look, just underneath where the 100% is, right? That blue line, that is from a bat virus, the sequence of which, the bat virus itself was collected from another area of China. The sequence was located at the Wuhan Institute of Virology. It is 96% the same as SARS-CoV-2. So the closest thing we've identified. If you look at that little red circle though, you see a dip. It's not a huge dip. It's not as big as the dips of the other viruses, including the original SARS virus, but it's a big enough dip that you notice it. And that dip happens to be right at the spike protein, right? So that's the most important part, and that's the part that's most different from the bat virus. So where did the gene coding for the spike protein of SARS-CoV-2 come from? Well, one theory is that it came from a similar coronavirus in pangolins. Okay, so pangolins are an animal that is traded on the illicit markets in China for medicinal purposes, my understanding. And the pangolin virus, which I don't have an image of here, so you just need to imagine it, is very dissimilar from SARS-CoV-2. Very dissimilar until you get to that same green spot where the spike protein is, and then boom, it's super similar, and then back down, okay? And most importantly, within that spike protein, that little green area there, there's an area called the receptor binding domain, the RBD, which is where the actual binding between the S protein and the ACE2 receptor on cells is. And in particular, that receptor binding domain is nearly identical, okay, between pangolin and SARS-CoV-2. Everything else is different. So one hypothesis is that a pangolin, which was infected with this pangolin virus, also became infected with a virus similar to the bat virus that we're seeing here. When those viruses are in the same cell and making copies of themselves, they make little pieces of DNA and then reassemble them. And those reassemblies can be confused. And one part of one virus can be reassembled with other parts of another virus. So it's possible, and the hypothesis is that during a single cell being infected with both viruses, the reassembly enabled the gene coding for the S protein to be reassembled inside the bat coronavirus. Okay, we don't know for sure, and we've never found a virus that has, or to my knowledge, we haven't seen a sequence or a virus that has those two characteristics, but this happens in nature. It's how we get recombination with influenza, and it wouldn't be unexpected, okay? That is the first big difference between SARS-CoV-2 and everything else, is that it's got this receptor binding domain that doesn't match the nearest neighbor, which is the bat virus. Now, the other thing that's different with SARS-CoV-2, and this time not from just the bat coronavirus, but from everything, is this thing called a polybasic cleavage site. So now we're talking about the lower left picture. And it really doesn't, I don't really understand a polybasic cleavage site. It doesn't necessarily mean, it doesn't matter if you understand it. All you need to do is look at the picture and see that in the area where it says polybasic cleavage site, the top line is SARS-CoV-2, and you see a bunch of letters. Everything below that, the first one being the bat coronavirus, the second one being the pangolin, and the third one being regular SARS, have a blank spot, right? Which means they don't have it, okay? This virus has it, it's different. They don't have it. What does this cleavage site do? Well, in order for S protein to bind, it needs to be activated. And in order for it to be activated, a protease on the cell that is trying to infect needs to cut it. And the polybasic cleavage site is where it gets cut. So if the cell is able to only cut at a single base, a monobasic cleavage site, like you would see with SARS-CoV-2 or even the bat, then you can only infect certain cells and maybe not very well. But here you have a much bigger opportunity to cut the virus, activate its receptor, activate its S protein, and enable it to bind. So most analysts are convinced that this has a lot to do with why SARS-CoV-2 is so infectious. And maybe we don't know why. There's no consensus on why. Maybe it's because it infects more cells in the body. Maybe it's because it infects better, but we think this matters. That is a type of mutation that we would expect to see accumulate over time, going back to the track changes analogy. As a virus is in humans, going from human to human, not very good at spreading, the virus is mutating, then one that spreads really well takes hold like a weed on the lawn, and because it has this type of mutation. The challenge is that this mutation was there from the very beginning. The very first cases we identified and the very first environmental samples that we have from the wet market in Wuhan all already had this, okay? So there's none of that expected lead up that we would think we would see. So how could that happen? And there are really three theories. The first theory is that an earlier version of this virus infected people much before we were ever aware of it and was slowly spreading, accumulating these mutations. Finally, it got the right one, and bam, it's December and we're off. That's theory one. Theory two is we still haven't found it yet. It's in animals somewhere. And there's something a lot closer to what we're seeing in humans now, and it evolved in an animal reservoir. And then the third theory is that performing a perfectly legitimate experiment, scientists were trying to recreate this process. And I'll try not to go on my soapbox because I hate these experiments, but the theory is that we know now people are moving into spaces where we're interfacing with animals and viruses we haven't before, right? So we go out and collect those animals and look at what viruses are in them to try to figure out what's out there that can hurt us. Imagine you collect 1,000 coronaviruses. Well, which ones do you focus on? One approach to knowing which ones to focus on is to do something called a passaging experiment, where if your initial studies and research tells you, hey, this thing could really do some damage in people if only it spread well in people, the next scientific question can be, well, let's see if it spreads well in people. And the way you do that is typically in a ferret, which has cells that resemble or replicate well what it would be like in a human. You would do what's called a passaging experiment. What that means is you take the virus, you infect the ferret, and you wait. And the ferret is sitting in a cage in a room full of other ferrets, okay, in their cages. And as the ferret becomes sick, you take back out the virus. Now this virus that you have now is different from the last one, right? Because it's been mutating. You inject that into another ferret, take it back out, inject it in another ferret. And over time, it's accumulating mutations, It's trying, or it's not trying, but through luck, these mutations are going to have an effect, and the one that's the best, like a weed on a lawn, will stick. Eventually, a ferret in the cage next to the one you're injecting becomes sick. Okay, that means somehow it got from the first ferret that you're injecting to the second ferret in the cage next to it, possibly through the air. Now you go to the second ferret and you take that virus and you see, okay, well, what changed from the earlier virus to this virus that let it spread from that ferret to the cage next to it? That's a typical passaging experiment. And so another alternative origin hypothesis for this virus is that the Wuhan Institute of Virology, which we know had these types of viruses, was performing this type of what we call gain of function experiment, which we know it does. And just to be clear, this could have happened anywhere. We do these tests in America. We do them with our colleagues in Wuhan, et cetera. So this is not about who did it. It could have been anywhere, okay? But it's possible that, took a virus, wanted to see if it could spread from human to human, did this passaging experiment, got one that had these mutations and had some sort of laboratory accident, whether it was an accidental infection of a laboratory worker, which we saw with the original SARS back after the outbreak. There were at least 2 other laboratory infections that led to clusters of cases in China. Or potentially it could be the virus was released in contaminated waste or some other option. So we don't know how the virus got so good at spreading person to person, but those are the sort of the 3 prevailing theories right now. The last one being, most people would say the least likely. How do we sort it out? Well, the best way to sort it out is keep collecting viruses and look. Keep collecting. Don't not try to do these crazy gain of function. Not crazy, but not try to do these gain of function experiments that are high risk. But keep collecting viruses and look for something closer. And that's in animal populations. And in human populations, go back and look at samples that were collected before all this started and see who may have had evidence of an earlier version of the virus. And most recently we learned that France had an infection in December because that's what they did. They took a bank sample of someone who had coronavirus-like illness, back then characterized as influenza-like illness, and tested it. And it turned out that it was PCR positive or nucleic acid positive for coronavirus. So those are sort of the two ways that we'll really be able to get at where this virus came from. Why does it matter? Well, in my opinion, first of all, it doesn't really. To prevent the future outbreak, we have to address these two problems, period. Animal-human interface due to human practices on a global level as a global community, and laboratory safety and high-risk research on a global level as a global community. But the reason we also want to know now is because if one of those possibilities, which is that there's another animal reservoir out there that is still holding this virus, if that turns out to be the reason it came to us so already well adapted, we need to know where that is. And we need to prevent continued reintroductions or, more mutations and reintroductions of different versions of the virus. Not that would necessarily be more dangerous because there's really no selective advantage in that, but because maybe our vaccines won't work against it. Or the things that are targeting, the S protein is where we target our vaccines, so the things that are targeting the S protein, we worry about mutations that could change what we're targeting, right? So that's why it matters, and I realize I probably talked way too long on it, but I just sort of have to go through it slowly to understand. So I think I had a couple of questions pop up. I'll just take a quick look. Friends had a question that I think is worth pointing out, which is, do the worst virus strains die with the patient? So that the idea he's getting at is what's called attenuation, where typically as viruses, and as we probably will see with this virus, ease into some sort of seasonal pattern, they become less dangerous but more spreadable. And it goes to, what is a selective advantage? It is not a selective advantage for a virus to die or to kill its host, because then it can't keep spreading, right? Viruses are like people. They want to make babies and see themselves, right? So, so there's no selective advantage in that. There is a selective advantage in spreading better. This virus has no selective pressure to spread better because it's burning all over the place. But eventually, as the susceptible population decreases, we may see a more attenuated form of the virus that spreads better, but is less lethal. And yes, friends, I think the most lethal versions of this virus would be expected over time to be outpaced by the viruses that spread better, but cause less lethality, because lethality doesn't help them spread, if that makes sense. Okay, next slide. All right, now we're finally to the part I think you probably joined for, which is where do we go from here? So these are 3 scenarios that were presented by a group called CIDRAP at the University of Minnesota. It stands for, I think, Center for Infectious Disease Research and Policy. Their director is an influenza specialist named Dr. Mike Osterholm. And he's famous for saying, I'm not trying to scare people out of their wits, I'm trying to scare them into their wits, right? So he's been talking about this concern for a very long time, and mostly fell upon deaf ears, unfortunately, and here we are. But he's an expert on this, and he also worked with another expert named Dr. Mark Lipsitch, who's a modeler and epidemiologist, and is someone that I'm sure you've read about in the papers, who's talking about what's happening next and where we expect this virus to go. So together they came up with 3 potential scenarios for what could happen from here. Their scenarios are based on how a virus behaves. I flip it to how we behave, because I really, and maybe it's just my own shortcoming, I want to feel like we have some control over where we're going. And as I worked through their scenarios, I thought, we really do I think, have control over where we're going. Again, we're worried about susceptibles and exposure. Okay, so working through these three scenarios, scenario one, which is at the top. Well, first, let's just get on the timeline. So in all three scenarios, you'll see an initial bump. Okay, that first little bump on the far left of all three graphs, that's the initial outbreak in China. Then you'll see a much bigger bump, which is where we all are now. And remember from our earlier slide, we're somewhere at the top of this bump, but it's plateauing and it keeps going, and we're not sure when it's going to start going down again. Okay, so there's scenario one, which is peaks and valleys. The concept behind that is virus enters a part of the population and burns through it. So you see this spike go up. And then as the susceptible population decreases, the virus goes down and it's at a lower level while it's spreading potentially to a new location. And when it gets to a new location and it hits their population, then it goes back up again and back down, and it keeps going every time it gets to a new location. So to me, the human equivalent of that is basically what we're doing now, where we have a number of different parts of America that are handling potential exposure differently, right? The states are all determining their own fate. And so while New York may be coming down right now, another part of the country may say, oh, look, at the national level, cases are declining. We can go forward and reduce our social distancing. Whereas really what they're seeing is a reflection of New York. And the minute they ease their social distancing, boom, it goes back up again. And oh no, we have to pull back, everyone go back inside, and it goes back down, and then somewhere else, up and down. So that, I think, is, that is, if we keep doing what we're doing now, probably the most likely future, in my opinion, is that we'll keep seeing this up and down wave. Now, option two, the natural version of this, why Dr. Osterholm and Dr. Lipsitch have it in here, is because this is the typical pandemic experience. And in the typical pandemic experience, say in 1918, we had this spring peak and then declined, and because of seasonal effects of the virus, where the virus doesn't spread as well because of it doesn't survive the environmental stressors as well, like UV light, et cetera. And people's host defenses aren't as challenged, right? Because they're not being challenged by colder weather and all the things associated with that. And people aren't in confined spaces, they're not all indoors, they're outside. You see the seasonal decline, and then winter comes and boom, because the seasonal decline, you've got lots of susceptibles, lots of chance for exposure, and then boom, the exposure hits and it's off to the races. And that second wave is so big that it overwhelms your healthcare system, and, Armageddon, basically, right? If there is any sort of bright spot with that, is that it gets it over with. So I would call that now that's the way the virus behaves. If we apply that to human behavior, that's sort of the throw your hands in the air, we can't take this anymore. Every person should be allowed to make his own decision, right? And as much as it sounds like I'm being facetious, I think that's a possibility that we might actually take some sort of approach like that, where like we tell people, look, you're all at your own risk. Go out if you want to, stay in if you want to, open your business if you want to. If you don't, fine, your problem. And then I would expect to see this massive spike, and whether that's economically driven or we need to get our kids back in school because they're really missing out on education, or we just need to get the parents back into the workforce, or it's football season, whatever it is, we really see that would be that sort of blowout spike. Now the third scenario is the rosy picture scenario, where I was really hoping we would be by the end of sort of our current shelter in place restrictions. And that's if we took federal government ownership. We said, we're doing it this way. We're going to find a way to move from a sledgehammer to a scalpel by testing, contact tracing, and then we'll be able to ease restrictions, at least on low risk groups, and move forward. So I'll talk through all three of those scenarios in more detail later, and I probably should have a clock in here so I know how long I'm dragging on. But those are the three general scenarios. At the end of all of this, no matter what scenario you're in, whether it's top or bottom, at the far to my right of the screen, you either achieve a vaccine that works, herd immunity, or there's been enough attenuation of the virus. Again, there's no selective pressure for the virus to attenuate now, but enough attenuation later after the caseload is such that it's harder to find susceptibles, and the virus drifts into a seasonal pattern. What we would expect is the seasonal pattern will be driven by how protective our immune response is, whether that's an immune response from being infected by the virus or from a vaccine. If it's based on so there are 4 other generally circulating coronaviruses that cause about 25% of our common cold. They're seasonal, and they provide about 40 weeks of protective immunity once you're infected. So that at a minimum would get us on an annual vaccination cycle. If we look at data from SARS virus or MERS virus, potentially we could have longer lasting protective immunity, but it's one of the big questions is how long will our protective immunity last? Okay, I'm gonna apologize. I'm not even gonna look at questions anymore because I realize I'm dragging on, but I'll just try to accelerate a little bit to get us through and have more time for Q&A. Okay, so regardless of scenario, let's just run through this pretty quickly. Regardless of scenario, these are the things we have to do. First of all, we need to communicate clearly. Someone like me, I have the hours that it took to go through and try to figure out how much testing and contact tracing we're actually doing. But those numbers should be up front, and the plan should be up front. And I don't think we even know what our goal is. You know, I know we're losing the game, but I don't know how we're even keeping score, right? So I think, that's number one. Second, we really need to continue to control mobility. Again, we had a number of introductions from China, even more from Europe. The majority of cases in New York came from a single case from Europe. The majority of cases in Louisiana came from a couple of cases in the United States. So cross-border travel is a real issue. Mass gatherings are another issue. The whole risk of infection comes with close contact, face-to-face, within 6 feet, for extended durations, and particularly in enclosed spaces. Right? So we do need to continue to limit mass gatherings. And then we really need to protect our high-risk populations. I'll go into some of the numbers a little bit later, but really it's anyone, I would say, from age 65 up and with certain underlying health conditions. We need to make sure they're taken care of, but that we also don't expose them unduly to infection. It's still very hard to find out how much PPE we have. PPE, first and foremost, needs to go to the healthcare workers that are managing this whole situation. And, if you read any of the stories, they're nightmarish. But second, to the general public. I would liken this to seatbelts. And a lot of you are probably younger and don't remember, but when I was growing up, seatbelts weren't a law. And when they became a law, it was not cool to wear your seatbelt. Like I was in high school, and if I had my seatbelt on, I was like, oh, look at that dork with the seatbelt on, right? But now, if I don't have my seatbelt on, I feel like I'm gonna fall out of my seat, right? So that's where we really need to get in our public mindset when it comes to masks. We have to wear masks. And to wear masks, we need to have masks that we trust, right? And I don't know if any of you have tried to buy them, but certainly they're hard to find. But then most importantly, we need the testing. We need the contact tracing. There are 2 types of tests that are really important. One is looking for nucleic acid, which is evidence of the virus. That's called a PCR test. The other is looking for evidence that the virus either is or was there by looking for antibodies. That's called a serology test. The PCR test typically comes from a nasopharyngeal swab, which is this horrible thing that goes into the back of your throat. But more recent studies have shown that saliva may be just as effective at detecting the nucleic acid. For serology, that's a blood test. So you need to give a blood sample. And there's a third type of test called an antigen test as well that's coming on board. But we need to get that testing up, and then contact tracing. A test doesn't do you anything if you don't use it. And using a test means isolate that positive case, find out the contacts, and quarantine them. How do you find out the contacts? Well, there are a couple ways. One is bread and butter epidemiology. You ask the person, and it can be a teleworker who is talking to the person. Who did you contact? Give me their names, give me their email addresses. I'll reach out to them. Another is to ask individuals or businesses, restaurants, to maintain a log. You know, it's not too much to ask people to participate in their own health and the well-being of the country. Keep a log, everyone you came in contact with. Oh, you turned out to be positive? Please share your log, okay? And then the third is to use technology. And the idea is that there's an app on your phone, and via Bluetooth, anywhere you go, if it comes into another phone, you'll exchange tokens. It will keep that token on record for a set number of days and delete it. If you turn out to be positive, you enter that in your phone, and it notifies all of the other tokens. Hey, you've been in contact with someone who is positive. Please self-quarantine. A test is in the mail. The test arrives. You spit into it. That gets sent back. And hopefully as short as 3 days later, you know whether you've been exposed as well, or infected as well. So that's the ideal scenario. But in America and in the EU, we've had tons of problems getting on that online, mainly because of privacy issues here. In Singapore, it's worked great. Okay, and then the last issue here is increased critical care capacity, which of course is if our hospitals get overwhelmed, you know, that right now is our biggest metric, right? We're trying not to overwhelm our healthcare settings, right? So that means that we should be looking at how many open ICU beds we have. And if we're going to exceed that, then we're failing, and we need to set our thresholds differently for letting people back out into the world. If we can add to that capacity by, say, converting parts of a hospital and then other things like we've seen on the news, you know, hotels or using public spaces or bringing a ship into your harbor, whatever the approach is, the more capacity you have, the more you can actually make a risk acceptance decision to ease social distancing, because you have the ability to care for people to the best of your ability, which means you have less preventable deaths. What we really don't want is a death that we know we could have avoided just because we didn't have the stuff we knew we needed, right? Less preventable deaths, and the whole pandemic shortens and we get through this better. So capacity is huge. And then there are other points here on what businesses could be doing to help the question of disinfection of spaces, which is a huge question, particularly in our food supply lines, where processing plants are part of the farm to fork continuum, and they typically have workers very close together in a cold environment, and it's ripe for propagation of the virus. And then, you know, just a last point, which is sort of obvious, but we don't seem to be doing a lot, is there's a difference between being inside and outside. We know that, right? So there's no reason why people can't work towards that type of difference and sort of strategize accordingly, whether it's allowing outdoor seating with with certain amount of spacing at a restaurant, or simply going to get your hair cut outside, as opposed to what I did, which is, you know, try to shave my own head and make an idiot of myself. Okay, next slide. So breakout on testing. I think probably everyone saw our President Trump say that the United States leads the world in testing, and that's true. That's sort of the middle graph there. The blue line is America, and we have done way more tests than everyone else, okay? But if you look at the line graph and the red arrow on the lower left of this chart, you'll see that per capita, our testing isn't in the lead. In fact, it's somewhere quite a ways down. So if our metric is total tests, yeah, we've done more than anyone. If our metric is per capita, we're somewhere in the middle. And then if you look at that global map, if our metric is total tests per confirmed case, we're doing really badly, okay? We have the most cases, and we're basically doing comparatively the least testing. So our goal shouldn't be to say, hey, we've done the most testing, because that doesn't help us. Our goal needs to be, we've done testing and it's been part of a plan, and here's what it's done to help our situation. So we know, based on what Dr. Fauci and other reports are telling us, we need more tests. What we don't know is how many more tests, and there's a range. I mean, I put one range in here that says up to 5 million tests a day. That's what Harvard University's Path Forward plan suggests we need. That's a different part of Harvard University than Mark Lipsitch and the, who I consider sort of a foremost authority on this. But nonetheless, it shows the range that we're talking about. And we're nowhere near that. And to exemplify that, here on the far right of the slide are 2 snippets from the same day's headlines. Okay, this is May 18th, earlier this week. US, big announcement, CDC is going to test 325,000 people, as if that's a big number. Same day, in China, they announced they're going to test 11 million people. We're going to test those people over the course of a year. They're going to test them now, okay? I'm not saying they actually are going to do that or whether they have the capability, but that's the disparity of thought that we're at least seeing in the public play out, okay? And testing doesn't matter unless you contact trace, but we need the tests, right? So once we have the tests, how do we contact trace? Well, we don't have enough people to do contact tracing. And again, what contact tracing is, you get tests, you find a positive person, you find out every person that person's been in contact with, and you ask them to quarantine while you test them, right? So the estimate for how many people that takes is somewhere 50 to 100,000 people for the whole country. Right now, we have a total of 11,000. California alone is saying they need 20,000, and the states are officially asking for 66,000. So we're at a deficit of maybe 50,000 plus contact tracers, and we're just slow to bring them on board. We have people that are out of work that are interested in doing it, and the job applications for the contact testing are through the roof, but we're a bureaucracy, and it's taking a long time to get those on board. I was really excited to see Congressman Mrna, for those of you who don't know, who represents the district where CNS is, along with other congressmen, introduced an initiative called the Health Force to address this gap. This was introduced just, I think, last week now, or maybe 2 weeks ago. And they recognized this issue and said, look, we need a standing force to get out there and do this. And I think that's great. In addition to that bread and butter epidemiology, and one more point on bread and butter epidemiology, that bread and butter meaning just people, how we've always done it. I know it sounds, or it probably sounds crazy to a lot of people who are saying, well, we're going to go out and trace, 1 million cases or whatever. Like, this thing is everywhere. We can't do it. Well, my first answer would be, that's what the shelter in place was for, was to ramp up this capability so we could get out in front of it. My second answer would be, look, we've done this before. There was a very devastating, human virus that killed 30% of the people that got infected by it, called smallpox, and we eradicated it from the planet. And that took us identifying cases, isolating them, identifying contacts, quarantining them, and vaccinating them. Okay, so this is a model that has been effective, but we believed we could do it. We had a massive healthcare workforce that did it, and it took a long time, right? So this is possible, but it requires full buy-in, and we really don't have that right now. The last note here on contact tracing apps, again, they would help a lot, and really to get where we need to be, we need to use them. But unfortunately, it doesn't look like it's working out right now, mainly because of privacy issues. So in addition, so those are the things that we need or need to do. And then here's just the quick list of things we still need to know. Okay, so first, we still don't know how much virus it takes to get infected. And that really tells us a lot about what the risk of different types of exposure are. So we could adapt our behavior if we knew that a little better, and we don't. Second, we don't know seasonal variation. We know with influenza, like we talked about before, typically you would see a 20 to 40% reduction over the summer months due to the conditions we mentioned earlier. I think it's like 20% in the New York area, or 40% in the New York area, 20% in Florida, because Florida's already warm, right? So they're already having some of that that environmental impact that reduces the ability of the virus to transmit. And coronaviruses, the other coronaviruses that are seasonal, sort of the same amount. When you say something is seasonal, that doesn't mean it disappears in the summer, it just means it goes way down and then it comes back up when the conditions become ripe for it to spread again. So in this case, we don't know, it's a new virus. There's a lot of people who haven't been exposed, but we're I would say, optimistic or hoping that maybe we'll see about a 20% reduction over the summer months. And that might enable us to open up our social distancing measures a little better. What is the school effect? You know, so many people right now, I've got, you guys know, you've seen the picture, I've got 2 little kids. There's this massive debate, at least in my community, about the risk versus benefit of school. How much is this going to impact 10 years from now when we study this? How much is this event going to impact our kids, right? Not just from a psychological standpoint, obviously, but from an educational standpoint. And we don't have any really good understanding of what it's going to do if we reopen the schools in the fall. The timing looks scary, right? Because it's when things are getting colder again. It's when football season starts. It's when, you know, the elections are coming, right? So, which I'll get to later, but that's something we still don't know. We still don't know how much of the virus is in the population. If you go out and just do these, what we call seroprevalence studies, where you ask people to check their blood for antibodies, we've seen up to 32% of populations have already been exposed to the virus and have developed antibodies. In general, I think we've probably got about 5% across, you know, on average in America, but you can see the different ranges there. How long does immunity last? We already talked about this. Other coronaviruses, 40 weeks about. For the seasonal coronaviruses. For MERS and SARS, longer. So if you get the virus, how long are you protected? If we get a vaccine that works, how long are you protected? We don't know. Does it help you if you were infected with the other seasonal coronaviruses? If you know, will that prevent you from getting infected with this coronavirus? We don't know that. That's called cross immunity. We don't know how effective our tests are. We think that they really have too high of a number of false positives, which will, you know, skew our results, and too high of a number of false negatives, both the PCR and serology, but in particular, the serology tests. We don't have any good treatments yet. Everyone knows the hydroxychloroquine story and the fact that, you know, our president's still taking it. The data aren't there, the safety is questionable. So that's sort of fallen off the plate of potential treatments, at least for now. A lot of hope was being put into remdesivir, which is an antiviral agent, and including by me. I was very optimistic that it might help. We conducted a clinical trial of the effect of remdesivir at the NIH, and what a clinical trial is, without telling people, you give one patient a placebo, something that is not remdesivir, it's fake, right? And you give another patient remdesivir. And the clinical trial was such that the patients with remdesivir had about a 4% better survival, which is not statistically significant. But nevertheless, it was significant enough that we ended the trial and said, look, we can't ethically be giving people a fake pill when we know that they would have at least a 4% chance better survival if they got the actual drug. So that's the right decision, it's the ethical decision, but it eliminates our ability to have good data. And then most recently, and I think it was maybe this week, we had a larger trial come out saying, you know, remdesivir does not have a statistically significant effect, protective effect. It does seem to reduce the time to recovery, but not statistically significant. So we really don't have a good therapy right now. Our main therapy for this is oxygen. And then for the vaccine, you know, around the world, we're testing hundreds of vaccines. At least 8 vaccines at this point, by my count, are in clinical trials, which means they're being tested in people. So typically you'll test preclinical, which is in animals, you know, mice or guinea pigs, and then you move into people. There are 3 phases. Your first phase is just seeing if it's safe, and if you develop antibodies that look like they would work against the virus, as well as what's called a T cell response against the virus. If that works, and that's small, then you expand to a bigger study called phase 2. And then your big dog, your randomized controls, your randomized clinical trial is your phase 3, and then you get approved. We're still following that process, but we're abbreviating it as much as possible due to obviously the concerns and the pressure, right? But the first vaccine to really enter the process is what's called an mRNA vaccine by Moderna and Moderna. And right now, we just don't know how effective it is, right? So the initial results look good. There's a big announcement earlier last week that, okay, we're moving into phase 2 and phase 3. Then some scientists came out and started questioning some of the data. Stock market went up, went back down, right? So we just don't know. And we're not putting all of our eggs in this vaccine's basket. There are advantages to an mRNA vaccine, which, you know, I'm not smart enough to explain how it works differently from other vaccines anyway, but the real advantage is scalability. You don't need to grow a whole bunch of virus and then either kill it or attenuate it like you do with some other vaccines. So you can reproduce a lot of it really quickly if it works. So, you know, we're hopeful that this is the one, but right now we just don't know, right? So these are all the big sort of unknowns. Okay, I think we only have 5 more slides, and I'll try to go through them quickly. So I have no idea what time it is, and I apologize. All right, let's go now through the scenarios. 1 slide each for the most part. And for the record, the hardest thing for me to do for this whole presentation was do that little gas can. So the current course is we're taking a gas can, we're dumping it, we're pulling back, we're dumping it, we're pulling back, we're dumping it, we're pulling back, in cases are going whoop, 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 up and down. Right now, what I've added to the picture on the upper left is a dotted line which says critical care capacity. In the current peak, I think, you know, it's fair to say we're right at that line, over it in some locations. And the goal is, as we go back down and come back up, we need to stay below that line. What does that mean? We need to define that line by location, right? So what is our critical care capacity in this city, in this town? And that will tell us when we can relieve social distancing and when we need to resume social distancing, okay? Without that information, we're flying in the dark, okay? What does this scenario look, how does it play out over time? What is the most likely outcome? It's gonna look different wherever you are. But a concerning finding, at least for me, and maybe it's just because I misread it the first time I read it, is a UK model that looked at this, and they found that it will most likely mean we'll be locked up at home for 2 months and then out for 1 month, and locked up at home for 2 months and out for 1 month. And that is going to extend into 2021 if we stay on our current course. Now, if we can somehow increase our ICU capacity, our intensive care unit capacity, we can let more people out because they won't be at more risk, there won't be more preventable death because we'll have the capacity to treat them, and we can compress the duration. But without having that additional capacity to treat, it's, you know, it's too great of a risk to let more people out using the current scenario. Scenario 2, this is the pandemic wave. What I said, sort of the throw your hands in the air, every man for himself, every woman for herself, go out. The picture is, you know, you're just dumping the gas can. There you go. You know, we're going to get through this. You know, whoever gets exposed is going to get exposed, and, you know, if they make it, great. So the real issue here is, well, are they going to make it, right? So that's the question, right? So about 80% of, and this is all in the box of the lower right. So about 80% of these cases are mild. I mean, there are tons that are asymptomatic, right? About maybe 2% of all the cases die that we know of, but we really don't know. But thinking back on where my mind was, I was of the mind maybe a month or 2 back where I thought, look, we know that the case fatality rate is much higher in elderly populations and populations with underlying health conditions than it is in the other populations. So that's the population we need to worry about protecting. And the other population, we really can use restrictions. My mind hasn't changed on that being the population that we need to focus on protecting. But the data are showing me that these other populations, it's not as simple as just saying, go out, you'll be fine. And the next slide sort of illustrates that. This is a just published study of a month of cases in New York City. Okay, so there were 1,550 cases, okay, of which 22% went into the ICU. Okay, the ICU being intensive care unit. So if any of you have ever been in a hospital, living or not, you don't want to end up in the ICU, right? It's scary, right? We're talking life or death situations, and 22%. Now look at the range. If you look at the graphic, the range of patients that went into the ICU. We're seeing 20-year-olds in the ICU, just like we're seeing 90-year-olds in the ICU. By the way, the reason the distribution is going up and down, it doesn't mean that 60s and 50s are at more risk of ending up in the ICU. What it means is they're the people that are out there in the world getting exposed more. They're the people that are getting infected more. And this graph is sort of reflective of that. But I wasn't expecting to see such high numbers going to the ICU in these lower age stratifications. And in particular, I wasn't expecting to see such high numbers of deaths in these lower age stratifications. So we just have to understand what we're talking about here. If we're saying go out, everyone has their own risk threshold, right? If I were still being an idiot like I was 10 years ago, I would just go and okay, I'll be fine. If not, whatever, right? But I've got kids, and I don't want my kids to be on this graph, right? It's not, there are data here, but it's a little bit of a risky endeavor for people just to say, oh, I'm young, healthy, I'm fine. We really are unable to predict who is going to have these bad results. We don't know if it's because they're having an overreactive immune response, or if they got just a big blast of virus. In other words, a big exposure of virus. Like that probably explains why healthcare workers who are often young and healthy are getting sort of severe cases. Well, first of all, they're stressed. They've been working all day long. They're dealing with all the psychological trauma. But second of all, they're getting huge exposures, right? So maybe that has something to do with it. We don't know. In a way, it's sort of like a rolling the dice situation. Okay, and now the most hopeful scenario and where we hope we would be, which is we take control, we increase our testing and our contact tracing, we go from a sledgehammer to a scalpel, and hey, if we can't get our numbers all the way down to what this graphic depicts, at least we can take, if you look in the lower sort of graphic, we can take that first scenario of these peaks and valleys, and we can make those peaks lower. And over time, as we get more testing and the case numbers are beginning to fall, then we can get more control. That is our best case right now, in my opinion. Now, there's a slide I had on how we bridge from now to there, but I won't spend much time on it. There's an article on the CNS website if you want to read it. But in general, the idea is, well, we don't have the tests, we don't have the contact tracing, and we don't have the healthcare capacity. So what do we do? And one solution would be, well, the fact is the United States as a whole does have tests and does have contact tracers and does have healthcare capacity, but it's spread out all over the place, right? So how do we target it to the areas where there's the outbreaks? And the argument for doing that is that if there's an outbreak in one place in America, or a cluster in one place, it will affect everybody, right? So someone in Atlanta doesn't, and I'm randomly picking a city, but someone in Atlanta doesn't want to say, it's not our problem. We got to protect our own resources. Well, by protecting your own resources now, you may have double the caseload later, right? So there is an argument for synergy geographically throughout the United States to do what we would call sentinel surveillance, look at people that are being exposed in the population. So for example, the wonderful Instacart drivers that come to our house, every 3 days with whatever crazy, organic stuff my wife orders, or, the postmen, or, the people that are performing essential jobs, or as a former colleague, David Hirschberg, continually points out, look in sewage, because we know the virus is shed in stool, and identify where the hotspots are, and then flex all of our, or, move all of our resources in that area and then grow our response that way. That's one way we could potentially tide ourselves over until we have more testing and contact tracing. So let's just go quickly through specific recommendations I would make. If my job were to sit and advise, the coronavirus task force, this is what I would tell them. First, you need a single voice. I say him because in my mind it's Dr. Kadlik, who again is head of ASPR. But we need a single voice and we need to defer questions to that person, and it just doesn't need to be so chaotic. Second, we need to know what our goal is. If our goal is to not to overshoot our healthcare capacity, then our metric should be critical care beds available. If our goal is to establish herd immunity, then our metric has to be antibody prevalence. If our goal is we need to keep from having preventable deaths, but we also have to keep from having preventable economic loss, then we need a metric, something like a population welfare metric. In Italy, when they were seeing tons of deaths, but mostly in their elderly population, I think the average age of death was 79.5 in Italy. I could be wrong. They started talking about having a life years lost metric, but we need to know how to keep score, and we don't right now. That number needs to be reported daily. It has to be at the top of every headline, as does number of tests and test results. We need to get the health force rolling that Congressman Panetta and his colleagues have introduced, which is fantastic. We need to work with the governors to do a phased reopening. The equipment and supplies, and in particular personal protective equipment and PPE and masks, we need to continue to get, and we need to get in reliable ways. It can't be the states are on their own. It can't be states having bidding wars against each other or the federal government. It needs to be coordinated at a national level. We have a mechanism to do that through the Defense Production Act. That's a very preventable problem that we could address at the national level. Next, important to me, seemingly less important to other people. I know we have the biggest problem right now, but we don't know how this is going to look down the line. We may be preserving ourselves later by having more exposures today. We have a role in the global community. We have for years now been helping and taking a leadership role, and it's our responsibility, the same way it is for states, to work together. If there are cases around the globe that persist, as the US gets better, those cases will ultimately be reintroduced here and cause another problem. This really has to be looked at as a global problem. So I would love to see us take more of a leadership role instead of, what I read in our World Health Assembly introductory remarks the past couple of days. Another big issue, the fall election season. Whether you're a Democrat or Republican, we all have to agree that voting is an integral part of who we are as a society. The Wisconsin primary in that box there, when they had open elections, they had a real problem. They had a major spike in cases, right? So I'm not seeing enough being talked about or done. And it doesn't matter whether I'm Republican or Democrat or what, but if I'm the administration, I would want to be advocating for safe fall elections as a priority. And then finally, if I'm the administration, I would want to be saying, look, this is the problem, and we see it and we're dealing with it, and here's the plan, go forward. But we also are aware that this happened under our watch and it's not going to happen again. So we have started a health force. We have started building these capacities, these American supply chains that will enable us to go from an initial stockpile to hold the fort to a scalable capacity to deliver supplies, deliver reagents, deliver tests, and we will not be in this situation. And then most importantly, we are going to work with our global partners to address the potential sources of the pandemic in the first place, not necessarily this one, but all future pandemics at the animal-human interface, and also the concern of global proliferation of laboratories and their safety and the research that goes on in them. So I'll stop there. I apologize if I rattled on. I really, again, don't know what time it is. I keep looking over my shoulders if that's going to help me, but there's nothing there. There's a window. So I will now defer to John and field questions.
Thank you very much, Rich. That was a very interesting talk. Dr. Potter, would you like to start the questions?
Okay, thank you. Just one question I've asked Rich in the past, and I haven't heard a satisfactory response yet from him or from anyone else. You know, we talk a lot about the asymptomatic individuals, and I'm still searching for some data on the characteristics of those individuals. I think when we last kind of exchanged views on this, Rich, you were unaware of any real data except from very, very tiny studies. And so I'm really curious, I mean, do we have a better feel for what are the traits of those individuals who may in fact be infected but don't show the symptoms? Because that would seem to me to be a very important finding, and I don't find anything being discussed, at least in the literature that I'm familiar with. So I'd be curious if you've come across anything that would be informative on that point.
Yeah, well, unfortunately, I still don't know really what those characteristics are. And I may misunderstand, but my understanding of why that's not routinely posted by CDC or otherwise is because of certain protective measures they take about demographics and things like that. But I don't know, and it's something that I'll dig into more and try to understand, you know, why some cases are asymptomatic, and maybe that's something that we can leverage. Sorry, Bill.
Rich, we have a couple of questions. We have one from Philip Bleek, and if you want to respond to that, we also have two questions from Michael Carboy, Avner Cohen, and Ferenc Dalnoki-Fares, and Professor Jeffrey Knopf. You want to take them? Or?
Yeah, I'll work from top to bottom. Can everyone see these, or am I the only one who can read them?
You can read them, yes.
Okay, but can the audience see them, or should I repeat them for them? So let's see. The first question from Dr. Blake. For those of you who don't know Dr. Blake, he's in our non-proliferation and terrorism studies area, an expert who supports CNS. He just did a great publication on acid attacks. Let's see. So how do we think about distinct motivations of avoiding overwhelming our healthcare capacities, avoiding excess deaths as we decide what our policies? So hopefully we covered that. You know, it depends on which scenario you pick. To me, avoiding excess deaths is a great thing that you mentioned, because that's what I continually fall back on is what is preventable. You know, in medicine, people die, right? But so the metric we use in medicine often is what are the preventable deaths? And I think that's a good metric to put here. So the next one is Michael Carboy. Are testing reagents an issue? You know, they were an issue earlier. They're a less frequently discussed issue now, but certainly as a preparatory measure for future outbreaks, we need to have a scalable and incentivized test reagent capacity so that we don't find ourselves in a situation where we can't do tests simply because we don't have what would normally be, you know, a common reagent. And for those of you who don't know what reagents are, there's your tests, and then there's a bunch of other little, you know, chemicals that you would use to say, prepare your DNA sample. And those things were flying off the shelves early and hard to find. So next question, also Michael Carboy, do you see private commercial businesses pushing testing, causing so much concern over privacy that tracing will be met with great cynicism? Well, I don't worry about private commercial businesses pushing testing. I want testing. I think that concerns over privacy about contact tracing, you know, that's why we're not able to do mobile tracing right now through Apple and Google, right? Because they haven't been able to resolve the privacy issues. So that is going to continue to be a problem. And it's unfortunate because that's an area that could really help us. And it's proven, again, there's an app called, I think, Trace Tracker that Singapore is using that's been really effective in helping them do their contact tracing. And then again, the point being, you find a case, you isolate it, you find the contacts, you quarantine them, you test them, right? That's how you get to a scalpel approach. Okay, A.B. Nurk at Middlebury. I'm sorry, I don't know your full name. Let me see. Seems we still do not know the virulence mortality rate, as we do not know the percentage of asymptomatic. That's exactly right. So we think the case fatality rate is 2%, but we don't know what the denominator is. How many cases are there, right? So maybe it's 1%, you know? Back initially, we were saying 1.5 to 3%. And I was saying, I think it's quite a bit lower than that because I think there's a whole bunch of cases we don't know about. I think we still are in a quandary there. Next from Friends, local question on contact tracing. I would love to involve students, especially Miss students who are excellent analysts. Do you have thoughts on this? Yeah, I do have thoughts on that. So contact tracing, you know, in the old days, or when I would do it, which would be overseas, not in America, you would go out door to door, right? Now it's telework. You know, you can sit in the safety of your home and do contact tracing. University of California, San Francisco has a contact tracing course. I think it's on 30 hours. So yeah, I would recommend reaching out to UCSF, seeing if we can enroll some of our students in their course, and then talking to Congressman Panetta's office about engaging them in the Health Force Initiative. Dr. Knopf, do you recommend that we all download one of the contact tracing apps? Yes, if there were one that everyone was using, I would think it'd be great. You know, for me, my wife would lock me in the garage for 14 days if I did anything like I didn't absolutely need to do. So we don't go out much here. But for anyone who's going out, and particularly anyone that's interfacing with other people, I think it's for your own safety, and it's for this, you know, the contact tracing app protects you if you come in contact with someone who's infected, but it also protects your friends, your family, the people that you come into contact with if you end up being positive, right? So it helps everyone. Michael Carboy, are there legal roadblocks, straight primacy on legal rights to having a federal response? So I don't want to go political here, right? But I think that, well, so public health forces are state-run forces, right? So your resources are really at the state and community level. But there's no law that the federal government can't lead a national coordinated response. In fact, I would argue that's been the expectation all along. So I do think that while the states do formally own their own destiny, I mean, President Trump can't tell states they have to open. States will make that determination on their own. I think that we certainly need a national coordinated strategy where we all do that as one, and not just say, hey, states do that on your own, so that downstream I have plausible deniability. It wasn't my fault. You know, I'm not suggesting that's the approach, but to me, that's the optics of it, right? We need ownership, and the ownership comes at the national level. Availability tests we did. Okay, one more from Dr. Blake. One thing that needs to be said, herd immunity via acquired immunity before a vaccine is available or if vaccine becomes infeasible is a plausible pathway, but comes at a horrifying morbidity and mortality. That's exactly right, right? So yeah, I don't want to give the impression that I think that the fact that we're continuing to consider easing restrictions and letting more and more people out to just get exposed is a good idea. I think we're making hard choices. There's no perfect solution. I think clearly scenario 2, where we just say everyone can go out, we want herd immunity. You know, my little description of the anticipated outcome there is potentially disastrous, and that's where we started talking about, you know, case fatality rates. Right now, our leaky lockdown approach is, you know, we're letting some people get exposed, so we're decreasing susceptibles, but we're also decreasing exposures to stay within, you know, our infrastructure constraints. I think, you know, that still is too many. You know, what we said at the very beginning, you know, these are real people. We need to get to the testing and the contact tracing so that we can get surgical. And that's the best way we can protect our US and really the global population. Okay, Sandy. Hey, Sandy. Dr. Spector, for those of you who don't know, used to direct our DC office and has recently retired and come back as a senior fellow. So it's great to see Sandy's name. New US cases are coming down, looking at 7-day averages. At the same time, testing is significantly increasing. Should we be measuring new positives per number of tests as a metric? Yeah, I think, so the new CDC guidance, which just came out 2 days ago, and I didn't even mention it, it's on an earlier slide. They named 6 gates that they want us to go through in order to consider reopening. Gate one is number of confirmed cases. That's a positive PCR test. Gates 2 and 3 are suspected cases, whether, gate 2 is whether they look like coronavirus but haven't been tested yet or haven't had a positive test yet. And gate 3 is whether they look like influenza virus, which looks like coronavirus, and haven't been tested yet. Gate 4, let me see, good thing I have it here. Gate 4 is, yes, to Sandy's point, a decreased number of positive tests. Gate 5 is the ICU metric we talked about, where you have open beds in ICUs, and gate 6 is an increased test capacity. So yes, we're not just looking at number of tests, we're looking at number of tests in the context of number of positives, and that really is what's giving us a more holistic picture of what's going on. The other thing I would say too, just to Sandy's point here, new US cases are coming down, but just so everyone knows, I mean, expect things to go back up, right? And we're easing restrictions now, and the science tells us that as we ease restrictions, people are going to be exposed, people are going to be infected, and we're going to see things go back up. So there's typically a 2 to 3 week lag between what we do and what we see. So easing restrictions now means things start to look worse again in June, okay? What else we got? Oh, thanks, Jill.
So, Rich, I have a question if you, well, actually 2 questions. You emphasized that face masks would be the new seatbelts. I like that analogy. But how effective are they other than the medical graded face masks? Because, we're all walking around with bandanas and old t-shirts wrapped around our faces. So, this is what the county here in Monterey has advised, anything from a face gated to an N95 graded face mask. So what is your opinion on that? And the second part of the question is about testing. Should we all get tested?
Okay, well, you'll have to remind me if I screw up your second question. But so face masks, what we know, right, or what I know anyway, is N95 masks are good both to protect yourself and the people around you. Now, that is different from the original narrative that we read about, where people were saying face masks really protect the people around you. That's not true. They protect both you and the people around you. They have a 3-part filtration mechanism. So there are 3 different things that go on that keep you safe. And those 3 together, 95 in the N95 means they're worst case 95% effective. At the worst particle size, they are only 95% effective. Every other particle size, they're even more effective than that, right? So we would expect those to be very, very good to protect people and the people they come in contact with, protect everyone, and those are what we really should be talking about. Surgical masks, I'm not so sure about, okay? Homemade masks, I'm also not so sure about. The biggest issue with those 2, I don't even worry about how much they filter. I worry about how people put them on and off. So what you'll see, at least here, is people will have their mask on and they'll walk up to you and they'll take their mask off and they'll put it back on. Every time you're touching the mask, you're touching what the mask is protecting you from, right? And that's how you auto-inoculate. That's how you infect yourself, right? You take the mask off, rub your eye, And so the whole idea is, first of all, you need the right mask, but second of all, you need a whole lot of it, right? And the average number of masks for a single case in a hospital is something like 500, and we have healthcare workers that are using 1 mask for not just that case, but for every case, and then they put it in a plastic bag at night and put it back on the next day. That's unacceptable for America or for any country in the world, right? And for these other masks, whether they're filtering effectively or not, people at least need to be educated on how to take them on and remove them carefully so that we're not actually increasing the risk. If it doesn't filter at all, and we're also now moving our hand to and from our face, touching something that is collecting particles, then we might actually be increasing our risk. And I don't think anyone has really, worked through that.
Thank you, Rich. That is not, doesn't make me feel so safe though. But how about the testing? I mean, you mentioned.
Oh, that's right. Second question. Yes, testing.
So here in Monterey, for instance, in the local, they encourage us to get tested, but testing is not available. They ran out of it. So
right.
What's your view? Should everyone get tested? Should we have minimal testing, targeted testing? What's your view?
Yeah, so right now, we need the testing capacity, we need the tests, and we need them administered safely. And once we have those two things, then yes, everyone should get tested. And it can be as simple as something mailed to your home. You can mail to your home and, hey, if we don't have enough tests, and we know that the highest risk of a single infection is to that person's family, you can mail me a container, I can spit in it, my whole family can spit in it, we can send it back, one test for 4 people, run a PCR, and we'll know not just whether I'm clean or whether my whole family's clean, right? So there's ways to expand our testing capacity without necessarily producing more tests. But yes, we need the testing. But right now, if we don't have enough, then it's hard for me to say, well, Jean, I know you've been at home, I don't think you need to run out and get a test, or myself, I haven't been exposed to anybody, so I don't need a test now, especially if we don't have enough tests, and especially if we don't know if tests are being administered safely, at, for example, a drive-through clinic or something, right? So it's on the government to get the tests out there and make them safe, and then it's on us to go and get tested.
Very well. I don't see any more questions. Bill, do you have any additional questions on your side? Bill, you're on mute.
Yeah, sorry about that. Thank you, Jean. No, I have questions that arise, every day, and I have the privilege of being able to ping Rich whenever I wish. But let me, on that note, again mention this feature that we have on our website now, which is the World War V, which has a Q&A component. And so if in fact any of our listeners have questions that occur to them after we no longer are in this webinar, by all means, send us, go to our website, raise the question, and Rich or one of our resident scholars who are expert in this domain will try to provide a response. So I think it's really an excellent feature, and it's worthwhile just to read the Q&As historically, because of many of the points that Rich some of our colleagues have touched upon in the past. So what I would like to say, I think this has been great. I think Rich really provided a tremendously informative and very systematic overview of where we are now, how we got to this place, and some sense of where we may be going in the future. And I want to thank him for taking the time to prepare an excellent presentation. I was delighted to see that I think at the peak we had about 52 participants who were involved here. I believe that John has recorded the session, so we'll be posting it and circulating it to other colleagues. And I think we continue to perform an important public function by making available these kinds of webinars, and we'll be doing more of them. But I want to thank you, Jean, for initiating the seminar. Thank Rich for his excellent presentation, and to thank our audience for asking excellent questions. And so I hope everyone stays safe, and we'll do this again in the not too distant future. So thank you all.
Thank you very much. And let me also thank Rich and Dr. Potter and everyone for attending, and wish you all a very happy Memorial Day weekend.